This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 239, recorded on April 1st, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
How's everything, Elio? You all well?
Fine. I'm still with it. I'm with it.
All right. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, and I'm happy that it's April.
Yeah, why are you happy? What's good about it? Just time moving on?
We're going to be able to spend more time outside without being all bundled up.
Yeah.
It's not March.
March is a tough
Or
month.
February.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
It's not March. March is a tough month, isn't it?
It is.
Not as bad as February.
That's true.
Is that the coldest month in Michigan, February?
Well, no, but it just feels like winter is getting a little bit long.
Yeah, well, not going to get any shorter, is it? With global warming. Well, actually, it will get shorter.
Let's hope not. Yeah.
Global warming. The mosquitoes will join us. Okay, let's begin with our presentation of interesting science, microbiology specifically. And let's start with a snippet with Michael.
So I'm going to talk about a novel aminoglycoside tolerant phoenix colony variant of Pseudomonas Originosa. So so here's the story. In the presence of an aminoglycoside antibiotic, tobramycin, under anaerobic conditions, Pseudomonas Originosa is able to transfer 8 neutrons from the element lead into the element titanium, which only has 26 neutrons, resulting in titanium becoming elemental copper. Well, remember, copper has 34 neutrons. And this then results in the lead being transmuted into gold because it has lost those 8 neutrons.
You sure you're not talking about April 1st?
Well, let me just finish my alchemist dream. This is just but one of the benefits with the metallic copper, adding to the synergistic action of this antimicrobial tobramycin due to the effects of creating metallic copper, which is in turn going after the microbe's electrons. So the microbe was fooled in thinking that it was inactivating the tobramycin, and it ended up creating a more potentially potent antimicrobial. The trick is that the microbe needs to be in the presence of lead and titanium and an aminoglycoside in order for this selection to work. But the microbe has finally accomplished the alchemist's dream of transmuting lead to gold with the help of an aminoglycoside. I knew microbes could move electrons, but the physics of moving neutrons, well, that is truly remarkable, which I only thought beryllium could do. So, as you can imagine, this was my attempt at an April Fool's prank. Not as plausible as Volkswagen changing their name to Volkswagen, but this paper is nevertheless as remarkable as my homage to all the alchemists and science fiction fans out there, especially Mark Martin, who is a quite accomplished science fiction author in his own right. So today's snippet is the title was right. The novel aminoglycoside tolerant phoenix colony variants of Pseudomonas Originosa. And it's from our friends at Ohio State, Seidekler, Moore, Wozniak, Anderson, Dusain, and Studley.
And Anthony Lee. Anthony Lee was a middle author there as well.
Oh, I forgot Anthony Lee. My microphone was obscuring that aspect. Yeah, thank you, Michele. And it was published in Antimicrobial Agents and Chemotherapy. So a little bit of background first before we dig into the paper and what a phoenix colony might be. We're going to start with calcium sulfate containing beads or cements or bone fillers that are routinely impregnated with antibiotics. And then these materials are often placed at surgical sites during orthopedic revision surgeries to provide local therapy to treat these extremely recalcitrant and often occult infections. That is, if you tap the joint and just try to see if there's any bacteria causing an infection, often those microbes fail to grow. So this is a really big problem. And in prosthetic joints are becoming more commonplace today, especially as our baby booming population ages. And recall that today's 70s are really the new 50. And today's aging adults are no longer intent with a rocking chair on the porch. They want to continue with their active lifestyles. So prosthetic joints like knees, hips, and shoulders are now more commonplace. And the scary thing is prosthetic joints like knees and hips, which are placed at about 500,000 joints per year, have an infection rate of about 1%. So that's really a significant infection rate. So now let's think about how the orthopedic surgeon are treating these infections. And as you can imagine, if they have antibiotic cements that are literally involved in securing the joint in the bone, as the antibiotic diffuses out of the cement or the calcium sulfate bead, it will diffuse at a defined rate based on the diffusion kinetics of the matrix that the antibiotic is encased in. And so as that drug elutes from that material, whether it be concrete, whether it be calcium sulfate, a gradient will develop, providing a range from a very high concentration immediately next to the material to a low concentration of drug, if you will, similar to what we see on those everyone who has ever spent time in a clinical lab has probably done an antimicrobial susceptibility test where you take an antibiotic impregnated paper disc, you drop it on a Petri plate that has already been planted with a lawn of bacteria, and then you see this zone of clearing, and you measure that zone of clearing with a ruler, and you then reference that against a known set of standards to assess whether or not the microbe is sensitive or resistant to the antimicrobial. So in this paper, the authors are talking about these little itty-bitty colonies that miraculously appear in this zone of clearing. And so what the authors are asking us to do is to change our perspective on how we view a bacterial lawn, and recall that in setting up this diffusion microbial susceptibility test, one paints a defined concentration on a plate. Typically, a McFarland standard of 0.5 is painted on the plate with a Q-tip, and a McFarland standard of 0.5 is actually a measure of turbidity, which translates into a concentration of approximately 10 to the 8th colony forming units per mil if we were plating E. coli. So then you drop an antibiotic impregnated disc, you toss the plate into the incubator, and then within a defined period of time, you come back and you look for an absence of growth or that zone of clearing. The trick with this particular study is here they paint and wait. They paint the Petri plate and then they wait for 24 hours. So that lawn actually...
Call it an agar plate, will you, Mike? Call it an
all right,
agar plate.
An agar plate.
It's not on the plastic, it's on agar.
All right. I stand corrected. It is a nutrient agar plate, and the medium that these guys are using happens to be LB. So the colonies are establishing themselves, and here's where we are suspending our concept of lawn and actually thinking about a biofilm. because the microbes are very crowded next to one another. They're effectively growing as a confluent lawn of bacteria or a biofilm. So it's a little bit different. But when they drop that calcium sulfate impregnated feed that has tobramycin embedded into it, the tobramycin diffuses out, akin to the way a paper disc will diffuse out, and it's actually able to generate a zone of clearing, just like we see a zone of clearing associated with the antibiotic susceptibility. Now, again, going back to anyone who's ever done susceptibility testing, they are specifically looking for these colonies that arise in this zone of clearing. These are often pinpoint colonies, very small, not very large, and they typically emerge at about 3 to 5 days after you drop the antibiotic disc. And so this is the focus of their study. They're trying to understand whether or not this is truly a new colony type. And obviously they think it is because they named it a phoenix colony. And they are contrasting against the 3 other colony types that typically occur under extreme selective pressure, like the presence of an antimicrobial. So the 3 colony types are, of course, classically resistant colonies. These are bacterial colonies that will look normal on agar plates. They will be luxuriant in their growth. They will grow immediately adjacent to the antibiotic source, and it's a completely heritable trait. Then we have what are termed the viable and non-culturable colonies, the ones that will just sit there almost with their arms folded, where they just won't grow at all. But then when you replica plate onto a plate not containing an antimicrobial, the colony will resume and grow again. And finally, there's this colony type that effectively grows in the zone of clearing. It's a pinpoint colony, and they want us to begin to go with them on their journey, trying to understand whether or not this is a truly unique colony or is it some variant of a resistant mutant.
And Michael, I really appreciated that they gave us a roadmap for this journey that we'll go on. They included a schematic, figure 8, which basically lays out how you can differentiate these different types of cell types that can resist antibiotics. So
absolutely, Michele. They teach the reader how to appreciate the remarkable data set that they are giving us. Paul Studley trained with Bill Costerton, and he is a very visual microbiologist. And if you look at some of the figures associated with this paper, they're quite remarkable in terms of being able to visualize highly metabolically active bacteria to ones that are not so highly metabolically active. And it's really interesting to see how they phrase their questions. Now, we appreciate that the pathogen that they are actually characterizing is Pseudomonas Originosa, which is a gram-negative opportunistic pathogen. And resistance to aminoglycoside antibiotics like tobramycin can occur by principally 3 different routes in Pseudomonas. They can make a better efflux pump to effectively pump out the antimicrobial. They can reduce uptake or decrease cell permeability. They can alter the ribosome binding site that the aminoglycoside interacts with. And finally, they can produce aminoglycoside modifying enzymes that will modify the aminoglycoside antimicrobial into something that it's not. So the question is this Phoenix colony got one of these heritable type trait. So one of their first really cool experiments is they asked the question whether or not the microbe is altering its pumps. And it's a really clever experiment in which they take an efflux pump inhibitor, which is a broad spectrum efflux pump inhibitor, and they put it into the Petri plates medium in concert with the tobramycin loaded bead. And what they were able to show is that these phoenix colonies still emerged with no appreciable difference in their number or their appearance or anything along those lines. So they conclude that the phoenix colonies are really not a lesion in the pumping system. And they also look at whether the persister cells that they were able to isolate persister cells from this modified bacterial lung. And once the antibiotic concentration dropped below the minimal inhibitory concentration, the persister cells were able to reactivate. And you might be wondering if they measured the concentration of tobramycin within the zone of clearing to answer the question whether the tobramycin has just simply diffused below its minimal inhibitory concentration. And that is the reason that could account for these pinpoint colonies coming up. And the answer there was no, because they were able to show that where the phoenix colony arose, it actually had 5 times the concentration of tobramycin that was over the minimal inhibitory concentration at the edge of the variant colony radiance. So this then led these brilliant authors to conclude that since the phoenix cells were unable to be properly characterized by known resistance or tolerance or persistence phenotype, it must be a unique but still tolerant phenotype able to survive and remain metabolically active despite the presence of high concentrations of antibiotics. And in fact, they actually give us a growth curve. They actually give us 3 growth curves, one of totally heritably resistant cells, ones that are sensitive, and ones that are in between. And the growth curves are literally on top of one another. Then they ask the next question is, could this trait breed true? And the unfortunate answer is that while the phoenix colony could arise in the presence of 5x tobramycin, when they would replicaplate it and then asked if that trait of tobramycin resistance was there, unfortunately, the phoenix colony resumed its sensitivity to tobramycin.
So that maybe is unfortunate for the microbiologist researchers, but it's fortunate for human health, isn't it?
Absolutely. Absolutely, Michele. And that's why the orthopedists are still able to rid us of these infections. But in their final set of experiments, they really begin to take Pseudomonas apart. And Pseudomonas is one of these peculiar microbes that we're always taught as an aerobe, but it's really not. Pseudomonas originosa is capable of a fermentative metabolism in addition to using oxidative phosphorylation to obtain its energy load. And Pseudomonas originosa can use an accessory electron acceptor like potassium nitrate, where the nitrate is then converted to nitrite. So it's effectively acting as, if you will, an analog to oxygen, taking waste electrons. And
Michael, isn't its nutritional flexibility one of the reasons why Pseudomonas is such a big problem clinically? It can show up and grow in so many different sites and conditions.
this is the scourge of the infectious disease physician, is the fact that Pseudomonas often causes skin and soft tissue infections associated with surgery. Any of you who have had your knee replaced know that it's a bloody operation. Your hip is, again, a significant operation where they do truly invasive surgery. And consequently, as they're closing, the Pseudomonas can literally set up housekeeping. So that's why I think they're going to this anaerobic experiment. And the bottom line here is anaerobic environments provide protection to the Pseudomonas originosa from the killing effects associated with this aminoglycoside antibiotic tobramycin. And this was a really clever experiment. They had two sets of plates. The first set of plates had the LB agar, and they incubated the plates in a reduced environment in an anaerobic chamber. And then another set of LB plates had potassium nitrate, the accessory electron acceptor, again in an anaerobic environment. They plant the lawn, allow it to develop for a day, and then what they do is they add the antibiotic bead. And what they learned is, in contrast to the complete clearance of the lawn on LB agars containing the accessory electron acceptor, so the tobramycin literally killed off all the Pseudomonas originosa if there was the accessory electron acceptor. But the lawn that didn't have the accessory electron acceptor, where we forced the Pseudomonas into a fermentative form of metabolism, where it's just literally eating the materials in the proteins associated with the LB, that lawn was active, and it effectively ignored the presence of the tobramycin. And so, supplementing with this terminal electron acceptor, the lawn was active and it was able to be killed by the tobramycin, even to a higher degree than the plates in the aerobic environment. They immediately took these plates, replica plated them onto LB with and without tobramycin, and after 24 hours, a confluent lawn was observed on each of the LB replica plates derived from the original plates lacking the potassium nitrate, and there was no growth on the replica plates containing tobramycin. So they offer us that this indicates while the anaerobic environment had protected the biofilm lawn from the original tobramycin treatment, the lawn had returned to the wild type levels of susceptibility once placed in an aerobic environment. Now, this is really wild from the perspective of the infection, because your joint is truly an anaerobic environment. Because remember, we carry around our oxygen in an iron box. We only let oxygen out to interact as a terminal electron acceptor. And for the most part, the oxygen is tied up in hemoglobin. So these replica plating experiments derived from the LB agar plates containing the potassium nitrate, they produce slight growth on the LB agar plate and no growth on the antibiotic containing plates, further confirming the killing of the original LB agar plate containing the potassium nitrate in the anaerobic environment. Collectively, these data indicate that these phoenix colonies may actually be protected by the anaerobic microenvironment within this biofilm, and that denitrification is not the metabolic process by which the Pseudomonas is effectively deriving the phoenix colonies to remain active and survive antibiotic exposure. And so they boil it down to this still, again, creates a challenge for treating prosthetic joint infections even though phoenix colonies emerge at a very low frequency, it's still possible that these then could survive antibiotic therapy. And oftentimes when you have a prosthetic joint infection, the antibiotic therapy goes on for many, many weeks to sometimes months due to the anaerobic niche that I think the microbes are finding themselves in. And so these phoenix colonies can then serve to recolonize contaminated joint and perpetuate an infection.
So I'm amazed that Pseudomonas, again, nutritionally very flexible. They show in their really nice flow chart in figure 8 that it has no fewer than 8 different cell types that can emerge and then exhibit antibiotic resistance, with this Phoenix type being the latest. I was also interested that it was aminoglycoside antibiotics that trigger this, but not other classes of antibiotics. So first, just to remind everybody, of course, the aminoglycosides are a natural microbial product. So during the course of evolution, microbes out in the soil are having to confront production of these antibiotics by their competitors. But it also reminded me of a paper in 2005, Lucas Hoffman, Sam Miller, and their colleagues published in Nature that aminoglycoside subinhibitory concentrations can induce biofilm formation by Pseudomonas and also E. coli. And it did so through a pathway that involves a second messenger, cyclic di-GMP. And they identified one of the response regulators that was critical for that biofilm formation in response to aminoglycosides. And they also showed that by forming biofilms, now Pseudomonas is more tolerant. It can protect itself from the antibiotic. So I'm wondering if part of this Phoenix colony development might also be related to cyclic di-GMP signaling and this other kind of developmental pathway.
They did not speculate about that in their discussion.
Right.
But I thought of that as well. And again, one of the members of their group is from the Department of Orthopedics. So it's clear that the orthopedic surgeons are really concerned about these chronic prosthetic joint infections that are very challenging to repair. Oftentimes, what they have to do is they have to remove the hardware, and you're left with an inability to walk simply because they've either taken out your hip or they've taken out your knee and put in a spacer. And so you're on crutches, which is really pretty debilitating until they can clear the infection away. And, you know, there's a lot of effort going on trying to understand this whole process. So I hope that these authors look for these other triggers to try to figure out if we can make perhaps an inhibitor of those cyclic nucleotides.
Yeah, it's very cool biology, and I would think it'd be right. It would attract a lot of interest from scientists, but also funders, because Pseudomonas is such a huge clinical problem.
Michael, would you expect this phenomenon to occur with other antimicrobials?
Well, I think anything that slows metabolism down. And so aminoglycosides can slow metabolism down, which is why I think they did the metabolic studies where they were showing that it didn't impact on the ability to grow. The Phoenix colonies had just a little bit slower maximum specific growth rate. The resistant colonies came in at about 30 cell division time of 30 minutes. The wild type came in just under 30 minutes, and the Phoenix cells were just above. They were growing under 20 minutes, or at about 20 minutes. And so, you know, you look at this and you look at the slope of the line and you say, well, is this telling us something? The peptidoglycan antimicrobials certainly interact with the cell's ability to divide. I would next look at gyrase inhibitors.
Okay. Yeah, they do make note that they tried some other antibiotics and did not see the Phoenix colony. So they saw it with tobramycin and gentamicin, but not ciprofloxacin or colistin.
Mm-hmm. Yeah.
So Cipro is a gyrase inhibitor, and colistin is a membrane perturbant.
Would you would you expect it with other bacterial species to occur outside of Pseudomonas?
Oh yeah. I would expect, because we see these tiny colonies coming up often in the clinical lab.
Interesting.
But they could be one of many different types of cells,
yes.
right?
Yeah. Again, they went to great lengths to prove that it wasn't just simply dilution of the antimicrobial, because, it'll diffuse away from the disc.
Or classic pumps that pump out the drug.
Pumps out the drug.
I have to say, there's a paper in the reference section from 1952, Letterberg and Letterberg. How about that?
Right.
Yes.
Replica plating. I just love when people, you
cite the masters.
Instead of citing some article 10 years later that used it, they go to the original. That's the way to do it, right?
That is indeed.
Esther and Joshua.
1952. It's lovely. All right. Thank you, Michael. Very interesting. Now we're going to head to the sunset where Elio is waiting with a paper for us.
All right. The paper is entitled Decreased Ecological Resistance of the Gut Microbiota in Response to Clindamycin Challenge in Mice Colonized with the Fungus Candida albicans. Now, if you understand the title, you understand the paper. But let me tell you, the paper is published in a publication by the ASM, the American Society for Microbiology, called M-Sphere. And the authors are Laura Markey, Antonia Pugliese, Teresa Tian, Farah Roy, Kyungborn Lee, and Carol Kumamoto. And except for one of the authors, that are all from Tufts University, and the senior author, Carol Kumamoto, is a member of my old department. So I have a personal liking for this particular paper. So the story is relatively simple. If you were to take mice and give them clindamycin, an antibacterial antibiotic, you alter their gut flora, the microbiome in the gut. You stop giving them clindamycin, they'll bounce back. And they'll bounce back pretty much to what it was before. And that is called ecological resistance. Okay? So you give clindamycin, you alter the flora, the fauna, the biota, and then you stop giving clindamycin and everything goes back to, quote, normal. However, if you infect the mice, or you allow the mice to be colonized with the fungus Candida albicans, that bounce back doesn't happen with the same alacrity, not quite as fast, okay? So what's going on? So the paper, this is relatively straightforward, except that the analysis of what's going on calls for a lot of bioinformatics. And this paper is worth considering because there's a lesson here, namely, no bioinformatics, no microbiology. It's very simple. For these people, young people who may be listening, this is not news. They grow up with bioinformatics, and good for you, because it's important. However, for the older ones, this is a little bit challenging because it requires an analysis known as co-occurrence network analysis. Now, for those of us who were around in 1952, as Vincent likes to point it out, that is Greek. It's essentially very difficult stuff. It is not to the people who learn this with mother's milk, and many know parts of it as being a principal component analysis. Those lovely graphs which sort related aspects out by clusters. So for those of you who know all this stuff, this is a piece of cake. For the rest of us, it's quite complicated. So what do they do? First of all, Candida albicans is the most common yeast or fungus in the human body. It is also a pathogen and a potential pathogen. It can cause all kinds of infections. In women, it causes vaginal infections. It can cause post-operative infections in a lot of people. It's an important component. So learning about Candida albicans via mice may not be the normal route, because mice are not infected with Candida albicans. Usually they have another kind of Candida, but it's a nice model system to study. So in order to get Candida albicans into mice, they have to give it an antibiotic, and the antibiotic is cefoperazone. I guess it's a, Michael, you would know something about it.
It's a cephalosporin. It's a beta-lactam antimicrobial class.
Anyhow, if you give mice cefoperazone in the drinking water for 10 days, you can then infect them with Candida albicans, and the Candida albicans stays there after you remove the antibiotic, okay? So it's an interesting system. The result is that if you do that, the microbial flora, which is altered during the treatment with this particular antibiotic, goes back to normal. Now the question is, does it get back to normal in the presence of Candida? And the answer is kind of yes. It goes almost back to the same bacterial composition as before, not quite exactly, but close enough. But the bounce back to the normal flora is slow. It's a slow going thing. So that's pretty much the story. The reason for using Candida, as I say, is that it's an important human pathogen. You cannot do these experiments in humans. You do them in mice. Mice don't complain. They have no way to tell you that that's not a good idea for them. Okay, so here is the idea is that ecological resistance is what happens after antibiotic treatment. The important thing of this paper is to understand how much bioinformatics it requires. It is really loaded. And for the, as I say, for the oldsters among us, this is a challenge. For the young people, it should not be. But make sure you understand that this is a lot goes into it. And the paper is loaded with material that deals with just this kind of stuff.
And by using the bioinformatics, they were able not to just list, here's who we see in the gut with and without the fungus, but rather what are the relationships? What is kind of the ecology between the different genuses of bacteria? So that's what they beautifully display in their
I
call
think
these diagrams. But
I get the message that this is good stuff. But I must say, I get the message by having to defer for the time being, a total understanding of the analysis.
I thought it was clever that they, after they saw that the community structure was different with and without Candida, they then challenged the animals with an antibiotic that put the community under stress. And because they did it at 3 different doses, they had some sensitivity, and they were able to find at an intermediate dose, the presence of Candida had disrupted the relationships, the interactions, the network, and therefore the mice microbiota was more susceptible to disruption by the antibiotic.
Let me tell you, two important organisms that were disrupted were Bacteroides, which is important in, for instance, in decreasing the incidence of C. difficile infection, or Akkermansia, which has a similar role. So 2 important species of the gut microbiota are affected, and that may increase the importance of this finding, the importance of what Candida does. Although grossly it does not alter the microbiome in detail, it does make a difference. So this is the story that Candida makes a difference in mice upon in terms of the response to clindamycin challenge.
What's the mechanism of this effect? Do we have any ideas?
Yes, no. Candida albicans increases the amount of unsaturated fatty acids.
Okay.
And that may have an effect on the microbiome,
right,
on the flora.
And remember from the last TWiM when we discussed the wonders of Akkermansia. Akkermansia lives at that mucosal barrier where the mucus is. And
it lives off mucus.
What Akkermansia is doing is feeding the intestinal wall those short-chain fatty acids that the enterocyte, the host cell, needs to survive. And the other caveat is that not all Akkermansia are the same. While it's the same by the 16S identification, it's COVID all over again. There are variants. And, it really is, folks have been listening to the story of the variants of COVID. And what we as microbiologists know is it's all about what the variant is doing for fitness for the organism. And so the Akkermansia community distribution is really, I think, part of the key. But the other thing that I think we're going to see in the years to come is our sophistication and our ability to do total genome sequencing of communities as opposed to just one gene will get better and faster and allow us to discriminate variant differences within a community to understand. And as Elio pointed out in the beginning, these are mice. We feed mice all the same food each day, and we're trying to extrapolate this understanding to what Candida could be doing in our guts, because Candida will infect our microbiome. And so I guess the real question, to reduce it to simplest terms, is the tweet that M-Sphere attached to the paper. Do bacteria care about fungi? and the tweet is really yes. The commensal fungus reduces the ecological resistance of the bacterial gut microbiota. So by virtue of the fact that the fungus is there, and the fungus got there because of the treatment of the cephalosporin antimicrobial therapy that Carol's team gave to precondition the model. That little trick that they did effectively answered the question, do bacteria care about fungi? And the answer is yes. And they begin to tease apart this beautiful network with all these interactive zones. And if you look at just the table, the tables are interesting in the sense that they give you abundance. If you look at Bifidobacter, you look at Sutterella, you look at Akkermansia, and you see the difference between uncolonized and Candida albicans colonized cohorts. And you see the numbers are different. And it's then they take us into this node centrality concept. And this is where Elio is trying to get us middle. I'm not going to call us old. I'm just going to call us middle people to begin to think like the bioinformaticians want us to do, to really understand that this is really microbial ecology that we're witnessing And we're using antibiotics to trigger these ecological disruptions.
In self-defense, I'll give you an example of one technique used. It is called fast, greedy clustering algorithm. I should understand what that means.
A simple way to think about it, for me at least, is each of the clusters that they display in their figures could be think of it as a tight food chain where the primary eater chows something and then lets off some crumbs, and then the next one eats those crumbs, lets off some new crumbs, but basically nothing goes to waste. So it's very stable and difficult for any invader to come in and set up shop.
Michele, did you connect with Laura Markey?
I sure did, yes. Yes, thank you. Thank you for the opening. So Laura Markey was delighted to hear her paper was going to be covered. She started listening to TWiM in college, but she got her start thinking about science. She said she was actually kind of bored by her high school science classes, but she loved books by Oliver Sacks, The Hot Zone by Richard Preston, Virus Hunters of the CDC by McCormick and colleagues. So she was really intrigued by microbiology.
Where did you go to college?
Well, I'm getting there. But her mother is just recently retired, but she was a registered dietitian, and she was a lecturer at the University of Michigan School of Public Health and also the bionutrition manager of the General Clinical Research Center here at University of Michigan. And she said it's from her mother that she learned what scientific research actually looks like day to day. So she got her degree, her undergraduate degree at Northwestern, and there she was working in Mark Mendel's lab. So he also is a ecologist. He studies commensal microbe-host interactions in Vibrio fischeri and the bobtail squid model. So she did some lovely work there. But when she was finishing with her undergrad degree, she was intrigued by working in public health and was able to get a fellowship from the CDC called an Emerging Infectious Disease Laboratory Training Fellowship, where she, for 1 year, was placed in the Massachusetts Department of Health. So it was a really invigorating work, very excited about it, but it did make her realize that she missed the lab and wanted to follow a research career. So she was admitted to Tufts' excellent microbiology graduate program, and she was excited to work with Carol Kumamoto because Carol was very open to the students investigating new questions and just kind of crazy biology. And in fact, her major thesis project, which she published in the journal Psychoneuroendocrinology, showed that Candida albicans actually perturbs the gut-brain axis. It can cause mice to become more anxious, and it actually changes their production of corticosteroid. Am I saying that right? Probably not. And if they pharmacologically reverse that impact on the neurohormone, stress hormone, they could eliminate the phenotype. So it was really beautiful work showing that Candida albicans is disrupting the behavior of its host. But this project, she said, was actually like a third project, but we can thank the pandemic that this came together because, as many people know, labs were shut down. And so you couldn't do wet lab research, but they had all this data that they'd been collecting. And this was an opportunity for her just to, sit at the computer safely at home and learn some new computational analysis pipeline, so the Q2ME1 pipeline, and come up with this more sophisticated analysis of the communities. She also something she enjoyed about this project is they were able to enlist some undergraduates who were doing summer research in Carol's lab. So I believe this would have been Teresa Tan and Kyungbom Lee, who had the awesome responsibility of collecting the mouse fecal pellets, from which they then did 16S analysis and began to study the community structure. So she is now at MIT. She's a postdoc, recently started with Tammy Lieberman, and is interested in setting up a mouse model to investigate host commensal interactions on the skin. And that's going to be another opportunity for her to dive deeper into learning genomic and metagenomic analysis that she'll need from her microbiota ecology studies. Laura and her husband live on the North Shore of Massachusetts, and she enjoys reading, cooking, hiking, yoga, and time with her cat.
Time with her cat. Thank you, Michele. All right, let me read one email before we say goodbye. This is from Nancy. Greetings to TWiM hosts. I want to let you know that I appreciate the work you do to discuss a wide variety of interesting microbiology topics in such a way that both emphasizes the complexity and interconnectedness of microbial life and interdependence of microbes and their hosts. In the summer of 2020, several of my microbiology advisees reported the loss of internship due to COVID-19 restrictions. Since I had used TWiM as a source of primary literature and a means to provide multiple means of expression, universal design, and as a source of primary papers to discuss in general microbiology, I designed a digital internship for students to analyze TWiM episodes and annotate them for use by instructors teaching general microbiology.
Wow.
The students listened to dozens of episodes and linked the papers and your discussions to the ASM fundamental statements used to teach microbiology. They also demonstrated science literacy skills as they had to summarize each podcast and justify their categorizations to their peers. This project was the basis of a paper that will be published in the Journal of Microbiology and Biology Education April 1st. I thought you'd like to see it before the special edition focused on the challenges of teaching in the context of COVID-19. I will likely host this internship in the future as the students were uniformly positive about the experience. Thank you for all that you do. And Nancy is an assistant professor at Iowa State University.
Oh.
And the paper, look, TWiM is in a paper.
That is awesome.
It's called Teaching in the Time of COVID, Creation of a Digital Internship to Develop Scientific Thinking Skills and Create Science Literacy Exercises for Use in Remote Classrooms.
Well, it certainly gladdens our heart. There's no question that we like what we're doing, but to find that it actually is useful. Wow.
Yeah, and a very
Not
creative
bad.
way to
Not
engage
bad. Thank you.
students.
Yep.
Yeah.
Yes, and that's the first research paper on TWiM. How about that?
We beat TWiM. We beat TWiM.
I don't know about that. I think there was one I wrote a while ago with...
Now, your writing, it doesn't count. Others have to.
Well, really?
Others have to.
Oh, you're making up the rules.
I'll make up the rules.
Okay.
We have to somehow
All right, that's TWiM.
beat TWiM.
TWiM 239. You can find the show notes at microbe.tv/TWiM if you want to send us a question or comment, TWiM at microbe.tv. And if you'd like to support us financially, microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you. And I will add to the show notes Laura Markey's favorite science-inspiring books that she read.
Very good. Elio Schachter's at Small Things Considered. Thank you, Elio.
I'm glad I survived this one.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.
As I say, I'm glad I survived this one.
Yeah, that was a tough paper, but you chose it, right, Elio?
I did. Little did I know.
Oh, my goodness. Yeah.
But I thought it was useful to bring up the issue of the centrality of bioinformatics.
Yeah, absolutely.